Today I'm going to talk about something that, we're all kind of very much at the forefront of our minds. And that is, how we're in the middle of this COVID-19 pandemic. What do we need to do in order to get back to our normal lives? So I would say that there's two steps that, we're working on and we've solved for, in some senses for this current pandemic, but there's going to be more novel viruses coming out and we need to find better solutions to make both of these happen. So the first step is we need to find ways to rapidly be able to make vaccines against new viruses, whether it's, new strains that are popping up that may have reduced resistance to existing vaccines, or just to completely, the next coronavirus that comes. And so many would claim that the vaccines based on RNA, so ones you've probably heard about coming out from Moderna and from Pfizer, that there are several reasons why these might be a very promising strategy to be able to rapidly make new vaccines. But the second step is that we have to be able to get these vaccines to where they're needed. And so that might mean having to be able to get these RNA vaccines to remote parts of sub-Saharan Africa, if that's where a new strain is, or it might mean having to be able to get global worldwide distribution. And there's been a number of questions that have come about for this topic with the rollout of these RNA vaccines. And so if you've been following the news, these distributors are requiring these vaccines to be shipped at these ultra cold temperatures. And the developed world has this is a prediction of just when widespread COVID vaccination will be achieved. There's a number of geopolitical factors in this, but a very real fact is that the developed world has been able to rapidly create new logistics systems. So here, this is a startup in Germany. They created these ultra cold shipping containers. In contrast, here's a picture of a containers developed that it helped end the Ebola epidemic in regions. It's called the Arctech. It was specifically engineered at one point to be able to undergo 1 day at minus 80 degrees Celsius. And so as a PhD student in a group who studies RNA, when we realized that there was this disparity in how RNA vaccines might be rolled out, we wanted to see is there anything we could do to try to engineer the part of it that we understand best, which is the RNA, to have a longer shelf life to better enable distribution of any type of RNA-based medicine. And so I want to start by just kind of a little bit of biology, reviewing how these RNA vaccines work. So you've probably encountered this kind of basic biological fact or kind of flow of information between these molecules. So in order to produce this final product of proteins, which are the main movers and shakers in the cell, there's two steps that take us from DNA being the genetic material in which all of our information is encoded. So first, there's a molecular machine called the polymerase that transcribes the DNA to make this messenger RNA. And then there's another molecular machine that translates this messenger RNA to then create the protein. I should say, my training was not in biology, so at one point I kind of had to understand this better by going and drawing some cartoons for myself to personify what's going on. So this ribosome is following a very complicated code that is encoded in this messenger RNA. So it's reading these sets of 3 bases and translating each of those sets of 3 to a particular amino acid that is then a building block of the protein. But there's a lot of redundancy in what RNAs can all code for the same protein. And this is a fact that's going to come to our advantage. So I want to now change gears a little bit and then talk about how does this process relate to vaccines, and talk about kind of the conventional way that vaccines are made. So, and I'm summing up, a huge amount of human discovery in some very crude cartoons, but basically what the conventional method does is we take an existing strain of a virus and they subject it to a lot of work and development to then basically make a weakened strain of that virus. And then it's that weakened strain that is grown and put in a vaccine. And so then when your flu vaccine in 2019, got injected, your immune system then is on the lookout and learning to recognize these dead or weakened viruses. In contrast, the premise of RNA vaccines that, a number over the last 2 decades, there is a number of just really inspirational scientists that have brought this technology to be. Instead of injecting an entire virus, they said, what if we can just find a way to just take the part of the virus that the body is going to recognize, which is this spike protein, and then just inject the RNA that is going to code for that protein. And then we inject the RNA, and then our body's own ribosomes are translating that RNA to then just create the spike protein. And so this was the hypothesis. And, we've seen now that these vaccines have, 95%, 99% efficacy, depending on what number you're looking at. So it's really a transformative way to create a vaccine, because now for any new strain or novel virus that is coming out, we can just very quickly find a way to create a vaccine against it. But there's a fundamental chemical problem with making any therapeutic out of RNA. And that is that it's an unstable molecule as far as molecules go. And so if you subject RNA to any amount of heat, or there's a couple of different types of chemicals, or UV, the message gets cut. And there's a specific chemical reason for this I'm not going to go into today. But this is the main form of degradation, and this hydrolysis is the name of the reaction. We wanted to see if there were ways to design molecules that wouldn't undergo this. And the trick that we have in hand is that, so we're used to thinking about DNA as base pairing. We think of this double helix, but it turns out that RNA, we think of it, you see it in pictures as kind of a noodle like this, but it actually can base pair too. And so it can fold back on itself and has the same GC, UA type of pairing. And the trick then is that there's evidence showing that when a nucleotide is forming a base pair, it's going to be less resistant to degradation. And so here I'm just showing this little cartoon of this structure I would call a hairpin. But really, these structures are quite interesting and really remarkable. And structured RNAs take on a wide variety of biological roles across the kingdom of life. And so I'm just showing here, it's easy to understand the base pairing of molecules. It's the subject of a lot of work to get these 3D structures, but I just kind of love them. And these types of structures are what me and my advisor Riju Das at Stanford University, this is what we kind of spend most of our time thinking about. And so there's just a lot of really fascinating biology related to these 3-dimensional structures of RNA. So back to this question, if we have a little protein, can we find a messenger RNA that codes for it, but that is more structured? So I'm going to start with just a little example before we get to something the size of a vaccine RNA that would code for the spike protein. This is a little protein used in biotech all the time. If you go and take that codon table and enumerate it, it has about 25,000 RNAs that code for it. So in my work, I developed a model to predict basically how paired or unpaired each nucleotide is going to be, and then relate that to a model we built to try to predict the degradation. So I'm going to basically kind of refer to this as the unpaired probability overall for the RNA. So if I go to a biotech website, there's a lot of tools that let me put in a protein and get back a messenger RNA to code for it. So this is a kind of a structure representation. So here I'm coloring each nucleotide by, in a calculation, how probable it is that it will be paired or unpaired. So these yellow regions, according to our model, will be more likely to be broken. But if I go through and look through all my 25,000, I can find another one that has roughly a 2-fold decrease in its probability of being degraded. And so that would correspond to this RNA having a 2-fold increase in its shelf life. So for this tiny little thing with 9 amino acids, we can go and just check all of them. But for the spike protein, there's a lot more. So it has about 1,200 amino acids in it. if I go back to a biotech website and do the same type of thing, this is the prediction of the type of molecule that I would see. You can see that there's a lot of these yellow loops, which are places our model indicates would be broken at. And for these 1,273 amino acids, there's 10 to the 633 synonymous messenger RNAs that are all coding for this protein. So, we can do our best to throw algorithms and stuff at this, but this is a huge space. And so, we're asking this question for this huge thing, what is the stablest synonymous RNA we can find? And this is kind of an open question. How much could we improve the shelf life of any vaccine just through this process of picking a different sequence? So in Riju Das's lab, we've found that when we have biological problems like this with just these huge search spaces, we have a lot of success when we turn it into a game and allow a lot of people on the internet to try to figure it out with us. So in 2011, Riju invented this game called Eterna. The idea is that there's a number of puzzles that allow you to learn principles of how different nucleotides fold. And there's a bunch of like funny little structures that you learn about. And so if I'm a player, I see this interface on my web browser, and my challenge for this particular puzzle is to design a sequence that folds to this particular structure. And over the last 10 years, there's been this round of crowdsourced scientific method system where players will then vote on sequences they think are most likely to do best at whatever the challenge is. Those winners will then get synthesized in the DAS lab at Stanford, and the results are then returned to players so they can see how their hypotheses did and learn from the results. And through this, there's just been a lot of really interesting developments in understanding how these RNA molecules fold. And so it was a mad rush of web development and science last year to try to then create games that teach players to design molecules that code for a particular protein. And what we found through a number of challenges that we put up was that players were able to find sequences that code for the same protein, but that show at least a twofold increase in this model's prediction for the lifetime. So here on the left, again, I'm showing what we get from this standard biotech method. And then here is RNA that had one of the lowest or the best scores under our model. It's made by a person whose screen name is Bugakman. Something that was really interesting. So at the same time, there were a couple other algorithms that existed for related things and also new algorithms that we built. And so we made a number of different types of proteins and we tested all of our design methods. And so there's a lot in this graph, but my main point here is that Eterna participants found the best score, the lowest score under our model for all of these. And this is something that I just think is kind of really remarkable in that, you hear in the news about deep learning methods like doing so well and deep learning has solved, this or has solved that. But in these new kind of very rugged problems that are at the kind of frontier of what we can design and understand, what we've seen repeatedly with Eterna is that human intuition, and not even expert intuition, but just getting people together who have different backgrounds to think creatively about new topics, they always win. And then as an RNA biochemist who has an interest in computation, then we kind of come along and play cleanup and try to systematize and build algorithms to code what human intuition has just seen right off the bat. So another really neat part of what Eterna players were able to accomplish is that these most stable sequences did not just have one particular structure, but they had a wide diversity of different structures. And this is really important because we don't understand, there's a lot we still don't know about how RNA structures interact with the body as a vaccine or in other respects. And so, for instance, here's this Bugakman superfolder that I just showed you, but other players also made much more branchy structures. So one of my favorites is this one from a player named Eli Fisker. He called it a Mandelbrot fractal. You can see it's much shorter and branchier, and it may be that there are other properties that this has that allow it to persist for different reasons. And so this is an open area of exploration for us. A final thing that we did that was really important for us. So these are predicted to be sufficiently stable that if we patch the sequence for different variants, those are still predicted to be as stable. So again, here's Bugak Man's design. I developed a way to go in and patch this to recode for all of these different variants. And so this is good news to us because it means that as strains come up, we don't have to ask Bugak Man to go back and redesign for all of them, but that we have heuristic ways to improve on these. So a question that we get a lot, and that is also at the forefront of our minds in trying to design these stabilized vaccines is, this model says that they should be more stable. Number one, are they actually more stable? But then can they actually then be translated? If you have this super compact structure, our little friend, the ribosome, still has to be able to hook on and translate the message. And so this is kind of an open question for everyone as well. And so with experimental collaborators at Stanford, we tested all of these things. So we actually synthesized some of the RNAs. We tested their stability just in syringes, so kind of to model the stability of a vaccine just while it's sitting in storage. And so this is a small subsampling of mRNAs that code for one of our model proteins that is fluorescent. So it's easy for us to monitor the fluorescent output that we see. So the in-solution half-life correlates with our model that we built. So for me as a computational person, that was very exciting to see. If we look at how much protein was expressed by these, there's not a clear, there's other interesting things going on here, but there's no clear correlation. But then if we look at a later time point as to how much protein is expressed, we saw also that there was a high amount of expression from these stabilized RNAs. And this is really exciting to us because it suggests that how stable these RNAs are is related to how much gets translated. So our kind of working model at the moment is that because the RNAs are able to just hang around longer, they can produce more protein.